Good afternoon, everybody. I'm very pleased to welcome you to tonight's ENLIGHT lecture series, Forest in Times of Climate Change. So, as you all know, forests are important. They mitigate the effects of climate change, but at the same time, they also are threatened and endangered by the effect of rapid changing climates and associated stress conditions. So we are very happy today to welcome two distinguished external lecturers that I will briefly present. The presentation will start with Dr. Sylvain Delzon from the INRA, the French National Institute of Agricultural Research in Bordeaux. Professor Delzon is also associated with the University of Bordeaux, and he is really leading in the research of plant ecology, physiology, and evolutionary biology, and combines these different disciplines with each other. The second speaker will be Professor Alireza Nakinizad from the University of Mazandaran in Iran, and also associated with the University of Ghent in Belgium. He is also a very prominent researcher in vegetation ecology, received a couple of important scientific honors and prizes, among them a distinguished award from the Iranian Academy of Sciences. So we are very glad to welcome these two researchers here. My role is to moderate the discussion. My name is Oliver Gailing. I'm chair of the Department of Forest Genetics at the University of Göttingen. So we will start today with a very short introduction. Hopefully I will restrict this to 5 minutes. Then we have the presentation of Professor Delzon and Professor Nakinizad, each 20 minutes, about including questions directly related to the presentation. And at the end, we will have about 15 minutes of general discussion. And then I also wanted to outline that there will be the opportunity of a network to attend a networking event starting at 7 p.m., to which I accordingly invite you all. So I will very briefly give a presentation just to introduce you into the topic, forest in times of climate change. We will hear about mechanisms of trees to cope with the associated environmental changes. And in the center of the long-term adaptation of this forestry population is the genetic variation as actually the raw material or the basis for adapting to stressful environmental conditions. And these stressful environmental conditions are related to an increase of temperature, more frequent drought stress events, but these kind of changes in environmental conditions are locally different and also different to predict. So here a brief summary of mechanisms that we will talk about. First, phenotypic plasticity, adaptation by natural selection, and migration by different means like gene dispersal by seed or dispersal of the male gamete by pollen. So first of all, briefly, what is phenotypic plasticity? This is the different reaction of the same genotype under different environments. So this can be, for example, analyzed in so-called common garden experiments that are established in different regions, and the different phenotypic response can be estimated at these different sites. So this is phenotypic plasticity. If we look at provenances planted at the same site under very similar environmental conditions, we can see genetic differences between individual plants and also provenances. And for example, here in Norway spruce, we see a very pronounced genetic difference among provenances in the timing of bud burst. We will hear about this later. So the genetic basis, when we are interested in the genetic basis of these adaptive trait differences, we have to generate both phenotypic data, so for example, data on the timing of vegetative bud burst, but also genome-wide information in adaptive genes to identify the underlying genetic basis of this. While we see very pronounced differences in these adaptive traits, like the timing of bud burst, generally, like forest tree population in temperate regions, show relatively low genetic differentiation at neutral genetic markers, which have nothing to do with local adaptation. And this is a reflection of like efficient gene dispersal, especially by pollen dispersal. However, like also rare long distance dispersal of the diploid embryo occurs. So this was just a short summary to remind you and to explain a little bit of these vocabulary that we will use also in the subsequent presentation. Just to remind you again, phenotypic plasticity, adaptation by natural selection, and migration and gene flow. These are mechanisms of plants and of trees to cope with environmental changes. So I will end my presentation here and thank you for your attention. I will give you the opportunity, of course, for a few questions that you can, for example, type into the chat. I will try to answer these questions. Are there any questions right now? Otherwise, I will stop sharing my screen. And I'm very glad to welcome Professor Delzon to give his presentation. So the floor is yours. Thanks a lot.
So I share my presentation. I hope you are able to see it. Is it working?
Yes, I can.
Yeah. So thank you for the introduction. I'm going to talk about the impact of climate change on forest and how forests can respond to climate change. So I will talk about genetic diversity a little bit, phenotypic plasticity also a little bit, but also I will explain how species can migrate. And as you know, a tree is a sessile organism that can only migrate via its seeds or pollen, as you already mentioned. And we also know that during the post-glacial period, species population responded to global warming by migrating toward northern latitude or elevation. And this results in local extinction that I call extirpation, you're gonna see, and also change in species distribution. So although the species migration has been recorded over the last few decades, in many cases, the current rate of global warming might be too rapid for natural migrations and to successfully deliver the species to the suitable habitats. So there is two main questions during my talks that I will try to address is, can plants and trees track current climate change? And what are the consequences of not keeping up with climate change? So that's the two main questions. So in my group, we try to answer this question that I can formulate in this way. Will natural population of forest tree species be able to respond to rapid climate change? And there is three main responses that we have been identified so far. So first, the species can migrate northward or upward. We can also observe some local extinction that we call here extirpation. And finally, species can also persist in current site through adaptation and through two different processes, namely here genetic diversity and phenotypic plasticity that you just introduced earlier. So I will try to illustrate those three kind of responses with some results we obtained in my lab during the last decade. And I'm going to start with the migration. So to illustrate this, this process, here I plot the map of sessile oak over Europe on the left, and you can see this is a current species distribution for that species in the blue points, the blue dots here. And on the right, you have like the prediction based on the niche-based model of the climate that are going to be suitable for the species at the end of the century. So in dark green here, it's where the species is not present right now, but where the climate is going to be suitable at the end of the century. And in a position here in red, you have an area where the species is currently present, but the climate won't be suitable anymore at the end of the century. A simple question is what will happen to trees first in this area, what we call here the leading edge of the species distribution range. Can we already observe some migration, or does competition slow down this process? So to address this question, we focus on several species, but today I will illustrate it with one species, Quercus ilex, Olmhawk. It's a Mediterranean species. And in the south of France, we have what we call the northernmost population of this species. All the model predicts that the climates that are going to be suitable for that species will move northward at the end of the century, as you can see on the last map here. So the species should be able to survive and colonize those areas in the future. So to check whether and to test whether it is really the case, can we already observe some migration for that species? We focus our study on the northernmost population that are growing here along the Atlantic coast in France. And we focus our study on four main forests, and we use a kind of retrospective approach based on the national forest inventories from the 1880 to date. So we have like a data set that is like a century and a half or something like that of present absence for that species. So what we found for the four forests, exactly the same pattern. So I will present only the two forests tonight here. So in green, this is the presence of Quercus ilex within that forest. It's 5,000 hectares of forest for each of the two forests here. So in 1880, where I am right now here, there is only few individuals of that species. And as you can see, the species spread through the landscape of that forest. And today, this is the dominant species of this ecosystem. And using those maps, we quantify the rate of migration of that species, and in average, we estimated about around 50 meters per year. So the main conclusion of this study is we did observe a migration of that species at the northern range of this species of distribution range. However, the rate of migration is much, much slower than it would be required to track climate change. So that was an example showing the species are able to migrate northward. And now I'm going to give like two examples about local extinction, here, what I call extirpation. So again, the same map, but here the question gonna be focused on the rear edge of the species distribution. So can we already observe some local extinction, some extirpation, or can the population adapt to the new climate or environmental changes? The two main questions. So to adapt to this new condition, there is two processes that would play a key role. So it's the genetic diversity of the population, or between population or within population, but also the plasticity of a given individual, of a given genotype, to express different phenotypes. So that's the main question. So there is two approaches that I will talk about today. The first one is the dendrochronological approaches that we use along an elevation gradient in the French Pyrenees here in southern France. On one species, Fagus sylvatica. So this is the current distribution of that species here on that map. And here again, this is the climate that should be suitable for that species at the end of the century. And as you can see, in southern France, the climate won't be suitable anymore for that species. So we will work on the mountain range here that is in the south. So we took cores from the trunk, and we were able to estimate growth. And using this curve, we were able to estimate the elevation for growth optimum. And we did that. We went back in time for different decades to check whether there is any change in the elevation of growth optimum, for instance. So here, to summarize it, I just plot two periods. So the ancient period in dashed line here, and the recent period in continuous line. So as you can see, during the ancient period, the population were performing better at low elevation, while at high elevation, this is exactly the opposite. The population are performing better right now. So at high elevation, where the temperature is limiting, climate change will have a positive impact on growth, where this is exactly the opposite at low elevation. And based on those two curves, we estimated the elevation for growth optimum. And here on the right, I plotted this shift in elevation that correspond to 30 meters per decade approximately. So that means this species shift its growth optimum upward. And we can expect to have like also a shift in this distribution upward during the same decades. The second example we can use is the observation of massive drought-induced mortality worldwide. And this is with a group of researchers, we quantify all those massive events that occur after a drought event. And as you can see here, there is even some drought-induced mortality in very wet environments in the tropics here, because a very short drought events can induce mortality because the species are highly vulnerable to drought. Those events, mortality events after drought, are mostly due to hydraulic failure. And I'm going to explain that. And I'm going to explain what we are doing to better understand the mechanism behind that. So hydraulic failure is when the air bubble is going through the vascular system of a tree, leading to desiccation and death. So we use two brand new techniques. One is X-ray microcomputed tomography at the synchrotron, for instance, technology. That allows us to get a scan of an intact plant and to check whether the vessels within that stem are functional or non-functional. When they are gray, they are functional, full of water, and when they are black here, it means they are non-functional, filled with air. So it means the air bubble spreads through the vascular system. That means that those vessels are not able to transport water anymore. So we use this technique, and also a brand new technique that has been published by a colleague in 2016 from University of Tasmania. This technique, what we call the optical technique, is based on the use of scanners. So we scan leaves every 3 minutes for many days. And we are able to see right in the middle here some cavitation events. It's the white lining you can see. When we sum up those events, we can map the spread of air bubbles through the leaf vein network. And so this desiccation process and the spread of embolism during drought lead to construct these beautiful maps of desiccation in leaves. So those two techniques, for instance, allow us to quantify and to characterize the cavitation resistance or the drought resistance of a species. So we use them to characterize the drought resistance of many species worldwide. So for many conifers, many deciduous species, and today I will give only two examples. One here, it's a study that we recently conducted on 19 oak species, Quercus species, growing in California. And the result is quite interesting. Also, we found exactly the same result within let's say the conifer taxa. So here, as you can see, there is a huge differences between cavitation resistance between species. So in blue here, the temperate oak species being more vulnerable than the species growing in very arid environments. And when we map this cavitation resistance onto the phylogeny here on the right, you can see that the species in purple are the more cavitation resistant species and are also the more recent species. So the main conclusion was that aridity drove the evolution of extreme cavitation resistance, and the species are adapted to the local environment. So there is these huge differences in cavitation resistance, in drought resistance between species. But within species, the story is totally different. So here I'm going to show like the result we obtain on two species, maritime pine on the left and Fagus sylvatica on the right. So for which we sample many populations through the entire distribution range. For the two species, we found limited variation or even a lack of genetic diversity for maritime pine here. For instance, we sample in common garden, but also in natural population, and we haven't found any differences in cavitation resistance between population, even if they are growing in a very dry environment in Northern Africa or in very wet environment near my lab in Bordeaux. For Fagus sylvatica, we did find some significant differences, but they are quite small, like 0.7 megapascal between the most vulnerable species growing in Northern Europe and the most resistant species growing in the south. So that means it seems that there is some genetic constraints for that trait. We can even call about like kind of canalization for this trait. That means that this trait cannot evolve within the species toward a more cavitation resistant xylem. So there is a huge variability between species, and there is almost no variability within species. And when we perform a kind of meta-analysis at the global scale, yeah, with some colleagues working in a hydraulic community, we collect data from 600 species from 4 different biomes, from very dry to very wet environment. And we estimated what we call the safety margin, so the distance of a given species to reach the threshold of hydraulic failure, so the risk of vulnerability under drought. And what we found is that most of the species operate very close to the threshold of hydraulic failure, like less than 1 megapascals. So that means that there is a kind of convergence in forest vulnerability to drought, and those forests will face some decline in productivity and survival if temperature and aridity increase in the future. So there was a two example to illustrate local extinction, extirpation, and I'm also working on tree adaptation to climate change by quantifying the non-neutral genetic diversity and the phenotypic plasticity, focusing on different traits, and mostly traits that are related to fitnet components, reproduction, survival, and growth. Try to quantify the adaptive capacity of those populations and the capacity to adapt to environmental change. Today, I will focus on two traits. The first one is leaf phenology. So in spring, we monitor during like almost 2 decades what we call leaf unfolding or bud burst. In fall, we monitor leaf senescence, and the difference between the two dates correspond to the growing season length. On the same tree, in natural conditions, but also in common gardens, we also set up some nets to quantify individual seed production for many years, a little bit less, but 15 years at least now. And the results show that for most of the species show a significant climb with temperature, showing that the leaf phenology shift with temperature along here the elevation gradient. But here you can see that Fagus sylvatica is the least sensitive species to temperature, while Quercus sessile oak, here it's the most sensitive species to temperature. This number means there is 6 days advance in leaf unfolding per degree increase in temperature. So 1 degree increase in temperature will result in earlier flush of Quercus by a week. So in conclusion here, this means also that some species will have some advantage under warmer climates and will be able to quickly respond to climate change, while others are less sensitive to temperature. If I focus on Quercus, Quercus petraea, the sessile oak, here I would like with this plot just to illustrate that there is a huge within population variation, and this is a very valuable resource for future adaptation. So here at each elevation, this is a population, and within a population, each dot corresponds to one individual. And as you can see here, the variability within the population is as large as the variability between all the population along the elevation gradient. So that means that there is a huge variability even if there is some local adaptation for that population. And this corresponds to a high adaptive capacity for those populations. So finally, we performed exactly the same approach with seed production here for 2 oak species over France. And we observed also that there is a significant climb with increasing spring temperature. So the warmer the spring, the higher the spring temperature, the higher the seed production for both species. So that means that climate change will enhance seed production for the oak species over Europe. However, we can discuss about it, but this might not be an advantage for forest regeneration for those species. We can talk about it later if you want. So to sum up, we evidence that there is already some migrations at the leading edge of the species distributions here for Quercus alex, but unfortunately, we have some evidence that the migration rate is much lower than the rate required to track climate change. So there is a need to study biotic interaction to understand why there is a discrepancy between those values. Finally, at the southern distribution, at the southern limit of the distribution range, we identify that those populations have like a high genetic diversity level And also, I haven't showed that, but there is a high level of phenotypic plasticity also, allowing the population to quickly adapt to environmental changes. On the other hand, we also found a lack of genetic diversity for drought tolerance, meaning that the population won't be able to adapt to a drier climate. And we also already observed some upward shift in southern France for some species. We are currently working on quantifying the pace of microevolution to know whether those populations will be able to adapt quickly to the current climate change. Thank you.
Yeah, thank you very much for this very nice and informative presentation. We have one question in the chat that I briefly check. The question is, did Quercus ilex spread without afforestation measures like planting or seeding? I'm not exactly sure whether this was from the audience, Jörg Ewald. Can you comment on it?
No, that is a good question. I did not spend enough time to explain everything, and clearly, Quercus ilex spread in response to climate change, but also to change in forest management. So in the paper, the conclusion was it's a shift due to global change. So we include climate change and forest management. So what's happened exactly? It's at some point, the National Forest Office has stopped removing seedlings from that species because there were like too many within the understory, and so it is clearly a response to both climate change and change in forest management. Yeah.
Perfect. Thanks a lot. There are no further questions. Maybe very short question from my side on the estimated migration rates. They were comparatively low. I think like 5 kilometers per year. And you mentioned the effect of competition. I think like the potential of trees to migrate, I mean, estimated in a completely different situation, like in tree-free regions after the last ice ages. I think it was like magnitudes higher, maybe like 70 kilometers. So can you comment on the effect of management and competition and maybe other effects?
Yes. Yeah, you're right. If you look on the kind of post-glacial migration rate that has been estimated in several papers, it's in between 100 meter to 1,000 meter per year. So it's like, let's say 10 times higher than what we observed here. First, we were working on a very small scale. It's 5,000 hectares. So we might have missed some long jump, for instance. So that's the first thing. And the second is during the post-glacial period, most of the colonization has been done in a kind of clear cut without any competition from other species. And this is clearly not the case right now. So Quercus ilex in the forest in southern France are competing with other oak species and also some maritime pine. So that might explain the discrepancy between the two numbers.
Okay, there is one other question from Professor Kotowski. Can you please repeat how migration rate was calculated?
Yeah, so we calculated from the map. And so there is different plots within the forest. So this is a 5,000 hectare forest with like 200 plots. And we use the center of each plot. When the species was observed, we calculate the distance in between the two observation time. And so we estimated the time of the speed of migration like that over the period. And we average it for over the whole period, actually, yes.
Oh, perfect.
So that means that there is some maximum speed that can reach like 100 meters per year during some, for instance, in 1960, the speed of migration was a bit higher than in 1880, for instance. But in average, it was 50 meters over a century and a half, something like that.
Perfect. Thanks a lot. We will have time at the end for some more discussion. And thanks again for the very nice presentation and the contributions to the discussion. So I'm very glad now to welcome Professor Alireza Nakri-Nezat, presenting on climate change impact on a very interesting ecosystem in northern Iran, the Haqqanian forests. So please, Professor Nakri-Nezat, thanks a lot for presenting.
So, is it working, Oliver?
Yes, everything okay. Perfect.
Okay, thank you very much. Thank you, Oliver, for your introduction. Good evening. I'm going to talk about a case study concerning impact of climate change on an amazing forest areas of Iran. Perhaps some of you know Iran as an arid country, but we have a marvelous forest, which I'm going to talk about. This investigation was done by a collaboration between our university and Lab group of Ghent University, and recently published in Global Change Biology. As you know, climate variables are the best predictors for species distribution models, particularly across large spatial gradients. This is a case for forest ecosystems and its understory composition, because understory species of temperate forests represent almost 80% of all vascular plant diversity. This layer of forest plays a vital role in ecosystem functioning and services. So any harmful impact on understory can automatically affect on whole forest ecosystem. Moreover, beside the climatic variables, it would be very important to test other non-climatic variables which are ecophysiologically critical for species distribution, like soil or land use. Incorporating different sets of environmental features along with climatic variables could make sure a more reliable and more accurate models of plant distribution. Among non-climatic variables, forest canopy cover is an important driver on species distribution models, because it determines not only the light intensity at forest floor, but also influence the microclimate of forest. So it is quite known that canopies mitigate global warming. Canopy cover can also modulate thermophilization, which is increasingly documented in ecosystems. Forest canopies can also an easy measurement for forest management. This is why canopy cover could act as a proxy for forest management. Actually, normally for climatic studies and species distribution models, having a macro data over a large climatic gradient, such as European scale, might be an important advantage. However, some exceptional sites have great potential for this kind of study. Our example site is Haqqanian forest, which are located as an arc along the Caspian Sea in northern Iran. This is dominated mainly by a deciduous old forest, and the forest is almost 850 kilometers long and has 1.85 million hectares surface. This is actually recently assigned as an UNESCO World Heritage and belong to, also more importantly, it's also belong to Caucasus biodiversity hotspot. And this area played as a refugium for a considerable number of arctotertiary elements during the last glaciation. More importantly, this forest is well known for its long-term continuity in forest cover. And according to historical accounts, logging was very limited before the 1960 or 1970 because of big road networks and forest management plans were almost lacking before this time. As I mentioned, the forests are located, the Hyrcanian forests are located on northern slopes of Alborz Range. Alborz Range is the largest mountain range in Iran. These are really unique in terms of vegetation in Iranian context, as it is highly contrasted with southern slopes, as you can see here in the southern slopes of Alborz Mountain, which face to the Iranian vast plateau. So elevationally, they distribute from only a few meters distant of Caspian Sea up to almost 2,800 meters, and with different bands of vegetation from lowland to upper mountain. So the vegetation range, like a more tropical form at the lowland to more open vegetation type at the timberline area. We believe that Hyrcanian forests have two important natural peculiarities that make them ideal location for niche studies. Firstly, these forests are a prime example of a mountain ecoregion with broad environmental gradients over a small and continuous area. Secondly, the forests are characterized with two important climatic gradients. One, as you can see in the arrows here, one from west to east, reflecting a gradient from a temperature, from a temperate subtropical oceanic bioclimate in the west to a more continental Mediterranean geyseric to oceanic bioclimate in the east. In addition, there is a 3,000 meter altitudinal range which reflects a decreasing of temperature and precipitation along elevation. So across these two elevational and longitudinal range, we have a considerable range of 14 degrees for the temperature. So we have a 14-degree differences, temperature differences from west to east, from north to south, in the elevational gradient. And also we have more than 1,000 millimeter precipitation range for whole study site. Interestingly, the temperature range here is almost equal to full climatic range of European temperate forest. So these peculiarities offer a good situation for testing theory of a space of time substitution. It means that this place could be a pilot study site for testing replacing time by space, because within this space you can test climatic change. So our research questions, actually, we want to say, we want to know how climate, soil, and canopy cover affect the current niches of understory species. And we want to know future climate change effect on potential distribution. of understory species, and how different scenarios of canopy cover change combined with climate change impacts affect the occurrence of species with different habitat requirements and different growth forms. To answer our questions, we had great efforts for sampling. More than 500 plots with 400 square meter surface were collected, including all vascular plants growing in each plot. We also have good environmental data, including soil, again, more than 500 soil samples. And also we extracted our climatic variables from CHELSA, and we also have, of course, our canopy cover data from each plot. As you can see here in the map, is the location of our study sites, and it also represents the temperature and precipitation gradient from west to east and from north to south. Out of all understory species, we only selected 95 species because these 95 species occurred at least in 20 plots. Our GAM analysis indicated that mean annual temperature and precipitation of warmest quarter are the best predictors in all species and in all studied growth forms. Before doing GAM, we had autocorrelation between all soil variables and all climatic variables to make sure that which variables are the best. This pattern is similar for 3 different growth forms. Also, the relative importance of precipitation increased for ferns. As you can see here for ferns, precipitation is more important for ferns, but it decreased for graminoids. On the contrary, the relative importance of mean annual temperature increased for graminoids, as you can see here, and decreased for ferns. And also canopy cover was the second most important variable for graminoids. But it is interesting that 4 edaphic factors we studied in our research they had almost the same mean importance after canopy cover and after, of course, after climate variables. At the second step of our research, we projected 2 different climatic scenarios with 2 representative concentration pathways, RCPs 2.6 and 8.5. Actually, these 2 scenarios denote 2 different amounts of temperature expectation by 2100. For these predictions, our soil variables actually kept at this mean values because we wanted to test our expectation from future climate. Additionally, we made predictions for both increasing with 50% increasing of canopy cover and also decreasing with 50% reduction of overstory cover, just to simulate the effects of management, forest management actually, based on combined effects of climate change and canopy cover, as you can see here in the different scenarios, with canopy cover decrease, canopy cover stable, canopy cover increases. And here you can see the different lines. Red lines indicates decline, and green lines indicates those species that are expected to increase. So, of course, it is more serious for ferns, as you can see here in the results. Particularly in a stable and decreased canopy cover, we have more serious effect on ferns, but they will be more successful when the canopy increases, as you can see here. ferns decline will be less. On the other hand, graminoids and forbs expected to decrease more when the canopy cover increases. At the next stage of the work, we also classified 65 species according to AUC. These species, we classified them according to ecological behavior based on expert knowledge in the recent decades in the Canyon forest. And we classified 8 categories, of which 5 categories of light adapted, generalist, thermophilus, shade adapted, and cold adapted. Because we only chose these 5 behaviors because they were more frequent in our species, in our 65 species. We wanted to see whether there is relationships between their probability of occurrence and different climatic cover scenarios. We tested these not only for plots in which the plants are currently available, but also try to model this for those plots that they are absent at the moment. Models show that even at the stable canopy cover, as you can see here, we have an increasing trend of light adapted and generalist, and also thermophilus species. And on the opposite way, the other way around, cold adapted, which are related to plants growing mainly in high altitude cold habitats, and shade adapted plants are expected to decline in future, as you can see in the red arrows here. So, but when the canopy is opened up, light adapted and particularly generalist and thermophilus species still increase. We have a more severe decline of cold adapted and shade adapted species here, like we had in our previous scenario. That means in a stable scenario. But interestingly, when the canopy cover increases, our models predict that light adapted, generalist, and thermophilus species will stop further increase. It is also the case for cold adapted plants. It means that a closed canopy could mitigate negative impact of climate change on cold adapted plants. So the canopy cover could buffer global warming against high altitude species. Surprisingly, it is very important result of our research. Shade adapted plants, as the most specialist of the forest, still decline despite higher, as you can see here, despite a higher canopy cover, and despite a more closer canopy cover, we lose a lot of shade adapted plants. This means that canopy cannot buffer global warming for supporting these forest specialists. And thus this group of species are potentially the most sensitive to future climate change. So for conclusion and take home message, I would like to give a short summary of most important results. We found even in the complex topography and remarkable variation in floristic composition, climatic related variables were the main drivers. Still in very complex forests, in very complex topography. And also we found future climatic scenarios showed that climate change are in favor of light adapted generalist and thermophilus species, but against mountain specialists. And we continue with forest management. Forest management, as represented by canopy cover here in our research, had aggravating and mitigating effects, different aggravating and mitigating effects on different species, depending on their ecological behavior and peculiarities. So keeping the canopy closed will mitigate the disruptive effects of climate change on cold adapted species. However, increasing canopy cover is not projected to buffer the predicted negative impact of climate change on shade adapted forest species, and thus this group of species could be potentially the most sensitive, as I said in previous slide. So the applied message of this outcome is that management regimes should be adopted to maximize biological diversity, especially in favor of forest specialists. The management could particularly crucial for Hyrcanian forest, because these forests are almost isolated and surrounded, as I showed you in the previous slides. This forest surrounded by a steppe vegetation. So any escape from the forest, any understory species escape from the forest will be surely fatal. For shade adapted forest specialists, or results imply a need to adopt other conservation measures in the face of future climate change. So this would be our main result. So any conservation procedure could take into account this important result. Okay, thank you very much for your attention. In my last slide, I want to show you about a recent discovery of that maybe, I'm not sure, but maybe it's the tallest oak in the world. It is Quercus castaneofolia. We recently discovered this tree in the Hyrcanian forest. It is about 60 meter tall and about 22 meter circumference. Thank you very much.
Yeah, thank you very much for this very nice presentation. We have a little bit time for questions, I think, but only like 2 to 4 minutes. I don't know, can we extend a little bit to address these 2 questions? One is again by Jörg Ewald: how variable are bedrock, geology and soil pH in Hyrcanian forest? Low importance for understory. Oh, sorry, that's now it went away. Low importance for understory looks unusual from a European perspective. Can you briefly comment on this?
Sorry, just I didn't get the question. Could you?
Sorry.
Or maybe I can check it from the chat box. Yes.
Yes, you can also from Jörg Ewald. How variable are bedrock, geology and soil pH in Hyrcanian forest?
Yes.
Low importance for understory. Oh, this low importance for understory looks unusual from a European perspective.
Yes, but actually in the current research, we didn't work on bedrocks, but it could be actually a nice idea to include bedrock as well. We only measured pH as maybe a proxy for different bedrocks. I mean, but anyway, we didn't work on geological bedrocks for or modeling, but maybe the model can be also improved by adding geological bedrocks. Actually, from west to east, we have different bedrocks, from siliceous to more calcareous from west to east. It's why that we have also different floristic composition. We checked if the soil could be a good driver for that, but we didn't find a good reason for to show if the soil variables could be good for modeling. But still, climate are dominant driver.
Okay, thanks a lot. There are two other questions. One is a bit long from Professor Konstantin Krutowski. Maybe the last part, I wonder how much Hyrcanian National Park is different from the area that you described in terms of biodiversity, altitude, and number of different ecozones. He visited National Park in Azerbaijan along the border with Iran, which belongs to the Caspian Hyrcanian mixed forest ecosystem. So how do these ecosystems differ from each other?
Yeah, thank you, Professor, for this question. This is very important question, but I wish I could also sample. We wish we could also sample Hyrcan National Park because Hyrcanian forest actually is from somewhere near to the border of Iran and Turkmenistan, and continue in the whole Alborz range and reach to Azerbaijan Republic. So, but we didn't have chance to go Azerbaijan for sampling. But I'm sure that we have many similarities. For example, we have Parotia persica forest and also Fagus orientalis beech forest in this national park in Azerbaijan. But actually in our sampling, we didn't have chance to go to Azerbaijan. And the area is called Talish Mountain. And it is only a portion, a small portion of all Hyrcanian forest. But maybe in future, we could add this portion in our study as well.
This sounds very interesting. A very interesting and important question from Esther von Richthofen. Thank you to all the speakers. Very interesting talks. One question to all speakers. Considering the changes you are describing in the three examples, will forests continue to be effective in taking in CO2, or will they become less effective or even emittents? Who would like to comment on this?
I can comment. So actually, the answer is going to be, it depends where you are within the distribution range of the species. And if you are on the leading edge or where the temperature is still limiting, climate change will have a positive impact on productivity, forest productivity. This is what we observed in northern Canada, in northern Europe, for instance. While if you are in at the southern limit of the species distribution range, so for instance, in northern Spain, northern Italy, for a temperate species, those species already experience a severe drought, and they're going to be negatively impacted by climate change, and they're going to be less effective for absorbing CO2 for CO2 assimilation. So yeah, it really depends where you are within the species distribution range. That's going to be my answer.
Thank you very much. Okay, Professor Krutowski has colleagues in Azerbaijan who would be interested maybe in collaboration. Maybe you can exchange emails later on. I think we are 5 minutes past time. I don't know how much we can extend. I think there are many important questions that we can't all address, but maybe in the following networking event, there are some possibilities to ask additional questions and discussion, since it's a very complex topic. And yeah, as Professor Delzon also pointed out, there seems to be high variation for some adaptive traits and for others no traits. So it's certainly important information also for forest managers to think about this, but I guess this would be another round of presentation and discussion to address all these topics. So we hope actually that we sparked some interest and some future discussion, and as pointed out, maybe also future collaboration, or at least like maybe some exchange of scientists when it's possible. So I would like to thank everybody again, especially the two speakers, and but also the contributions from the audience and the discussions. So there will be a link that can be sent by Alexandra Boat to everybody. I think it has already been sent, so that we can access this virtual room for the networking. Is this somewhere? I think it's somewhere available, maybe also in the chat. Is this correct? So with that, I think we have to say goodbye at that point, and thank you very much again. There are actually the links that you can also follow, so you are very welcome to join our virtual networking event, where you can also visit virtually our university. So hope you welcome you there. Thank you very much. Have a nice evening, and it was really a pleasure talking and discussing with you. Thanks a lot.
Thank you for the invitation. Thank you.
Thank you very much.
Take care. Bye.
Bye.
Bye.